we seen a
Right
lot
at
of
the end
shrimp
here.
or anything else in the
you can
water?
kind of go
Oh,
laterally.
not so much.
We're
I know.
comfortable right now, so we'll come down a couple meters and
The fish ate them all.
give you a little more playroom.
Yeah, you got a good 4 minutes till that next move starts.
Okay. You want to turn the lasers back on?
Sure.
Yeah.
Lasers on.
What is that sort of bluish looking thing? Center right of
The bluish looking thing?
the...
Oh, I see. It looks like it might be a holothurian. I see what you're
maybe,
looking
yeah,
at.
it's just
Yeah, right
above
there
the
by the
lasers,
lasers.
right?
Yeah,
Yeah,
might
that
be a
looks-
holothurian. I'm
Yeah.
not sure.
We've seen a lot of these guys,
I'm
too.
not sure. But
Do
we
a
can
quick
we can
zoom.
do a
Watch
quick
your
zoom.
toe.
Yeah.
Okay.
Sure.
Yeah, just see what that is there. Yeah.
Oh, gosh. Looks like a lot of manganese nodules in the sediment
It sure
ponds
does.
here.
It sure does.
Come ahead a
So
little bit.
both the crust
Is
and
that anything you're
individual
interested in there,
nodules
Watch
can
Lee?
be
Yeah, just tugging at each
biologically
other now, so-
mediated.
Okay.
In
Yeah.
other words, bacteria
Couple
or
minutes.
other animals too can interact with the sea floor and with the water column,
Look at it.
extract ingredients from their constituents that help them to metabolize, and then they will leave behind a concentration of a whole variety of different
Okay, so
metals
then-
and other elements.
Okay.
Is that one we've seen?
Yep, that is one we've seen several
okay.
of.
That's very cool. You can
Beautiful
see
animal.
see its gut.
You can see it moving
It's moving.
its right hand, upper right hand end, which is probably the, I'm not going to say it's the mouth end. Kind of looks like it has the
gut
mantle.
loaded with sediment. Sure does.
Okay, is that enough
There
of
you
that?
go. Yeah, that's good.
Okay.
Yep.
All right.
Thank you, pilot.
Just look around.
Just
So
another
at the
minute
There
or
depth-
are a
so.
lot of nodules here.
Yeah, there are. At the depth that we
Watch your
are
port
at,
side.
Yep.
the pale white specks you see in the sediment are likely the shells of these little one-celled organisms,
There's a big-
foraminifera.
Yeah, I see it. I see it.
Okay.
Their shells are called tests, T-E-S-T, and they are minute little coiled. They almost look like a gastropod, some of them.
So we've had a question about whether or
Oh.
not we can age date the sponges. Do we know how old they are? Do they have calcareous parts? So
All right,
actually
let's move
there
in
are
now.
only a couple groups of sponges that have calcareous parts, and neither of them live in the deep sea. So there's a whole class of sponges called the calcarea, but you don't find them in deep water.
So yeah, about 4-minute delay.
Because calcium carbonate doesn't do so well in very deep water.
No.
And then there's another group of demo sponges that secretes a calcium carbonate skeleton a lot like a coral. This is a big Walteria glass sponge, as well as siliceous spicules. So most of the sponges, in fact, all of the sponges that we've seen on every single one of these dives all produce siliceous spicules. So they're made out of silica. And they belong to two classes of sponges, the the hexactinellida and the demo sponges. Mostly we're seeing the hexactinellids or glass sponges. But to answer your question, very, very hard to date them. We don't know how fast they grow, how long they live.
Oh,
it has been, the only
Oh,
ones
I
that
thought you were
are
talking about the
easy to
rock.
get to date are the ones that produce the calcium carbonate skeleton like corals. And those are sclerosponges, and they formed reefs.
So watch it over there. I thought,
Yeah.
Actually,
Yeah.
they're still
It'll be okay.
substrate builders in places like Jamaica.
Okay.
But they're slow growers, and even something that's small, maybe a few centimeters in diameter, might be hundreds of years old. It's much more difficult to guess what the age is of these deep water
Yes.
sponges, and in fact shallow water sponges as well. It's been estimated that some of the barrel sponges that we see, for those of you who dive in
So 0.2 knots, 30 meters
shallow
is
tropical
like
waters,
4
those
minutes or
big
something?
barrel
Is that
sponges,
right?
it's been
5 minutes.
estimated
5
that they
minutes.
could be hundreds of years old.
Okay. So I get another.
It's about, well, in about 2 minutes here, we'll call it
Yeah.
another one,
Yep.
according to our little record here.
Yep. So in about.
In fact, I think one estimate
minutes,
even placed one
we'll
of
be
them
done
at
with that
about
move. That's perfect.
2,000
Yep, that'd be a good pace.
years old.
So basically, it means like 5 minutes of moving and then 3 to 4 minutes of stationary.
So
Wow.
this is
That's
a
a good
markedly different area. The substrate is
That should be
different.
enough
It's
to get us to the
more
waypoint.
nodules.
More highly sedimented is part of
it is.
part of the reason we are
Coming down a
on a bit of a step now. We've come up kind of a steep portion of the slope and are getting to, well, getting
does the
closer
auto
to
depth
anyway
work
the
or auto,
the
no,
top
there is no off the
of this
bottom.
ridge that
Auto
we're
altitude.
climbing.
There is
So
auto altitude.
Does it
are
work?
we one,
Not-
two,
not
three,
not very
halfway
well.
between 3 and
That's
4?
part of that
Waypoints
DEL
3 and
system.
4?
Yeah, and so
partway
you're
between,
it's in
Partway
the middle of the
between
vehicle.
waypoints
maybe
3
a
and
third of
4.
the way up
Okay.
from 3 to
It's just
4.
not a
Okay.
reliable way to
At
fly.
most.
Okay.
But it is getting, the slope is getting shallower here, and as a consequence, more sediment will collect, and then more of these little nodules form, and they're sort of pebble-sized, maybe, size of an egg at most. Many of them are smaller, scattered all over the surface in what otherwise would be sediment patches between the small boulders that you see sticking up off the seafloor.
So we had another question about how old these sponges are and whether or not we could use the tissue, the cells, to date them. Again, that's very, very difficult to do. As I know Deb knows this, I've been working on sponge cell culture for a long time and trying to get these cells to divide. I often joke that they divide on a geologic time scale, but I think that's just because we're not giving them the right conditions that they need to
Yeah, I was going to
divide and
ask you
grow.
that.
Yeah,
Yeah,
I'm going to go ahead and call that next
If
one.
they're happy
But
in
there's some-
captivity.
Sounds good.
Yeah,
Yep.
well, some of them are,
British
but they divide it. It's really species dependent.
move,
So there's some that we
same,
see in
move,
relatively
surprise.
unstable environments, especially in the shallow water where you'll have wide
Good diving.
fluctuations in temperature
Yes, you
and
gotta-
salinity.
3 or 4 minutes.
Okay.
That
Move
are what we
laterally
call
and just
ephemeral
it'll be fine.
species. So they'll grow really quickly, last a season, and then die back. So those are easy to monitor because we can do that because they're really in shallow water, sometimes just in snorkeling depths. But for these deep water sponges, we just don't know. To the best of my knowledge, and Chris, you can correct me, I don't know that anybody's kept any of these alive for any length of time. Sally Lees, I know, has done some work on hexactinellid sponges, and that's because she works in an area where you can get these glass sponges in relatively shallow water. So with the exception of some fjords around British Columbia and in Norway, where you can get these glass
After this
sponges
move,
at
it's,
much shallower
probably
depths,
changed
within
to 060 to
scuba
kinda
diving
start
depths. Most of them
easing
occur
our way
deeper
up
than
there.
200
Towards the
meters.
endpoint.
Yep.
But they were interested
Has
in
anyone
staying along
made
the edge of this.
time
I guess we
series
could do that if
studies
y'all would rather go along.
of sponges or particular
talk to them again.
sponges in a given area to
Because then we can cut
monitor
north right at
growth?
the end.
You know what? I'm thinking
Just go
about
right
that,
here, and then
Patty, and I
head more
don't.
north.
You know what? I don't think so. And I tell you the
050
reason I don't
towards the
think
end.
so is because we have very limited opportunities to go back to those same areas time after time after time.
Even
There's-
even in
065
scuba,
average.
Scuba depths.
Yeah, I mean, we can
yes.
kind of feel it
Scuba
out
depths,
as they
yes.
make these hops.
Yes, as a matter of fact,
That's what I was
Janie
wondering.
Right now,
yes,
it's okay,
Janie
but
Wolf
might find that it shifts
at,
more north
at
once we
Florida
move up
State
ahead.
University has been working for decades
I think they're
on
interested in these
sponges
really
in
high
shallow
biological
water
communities at
environments,
the
and
edge of
she
the ridge.
monitors them. She's been following like hundreds of individual sponges
Right.
in the Caribbean, so she can see what's happening in terms of their growth, death, what happens when we have severe storm events and they fragment off, and really, really interesting. So again, very much species specific.
right?
Yeah.
Okay.
But for those of you who might be interested, I'd really suggest that you take a look at the literature for Janie Wolf, W-U-L-F-F. She's been doing this work for decades, mostly in the Caribbean. Another person researcher from University of North Carolina, Wilmington, is Joe Pollock. And Joe Pollock has also been working on sponge communities in the Caribbean for, I want to say, for decades as well. So check out some of Joe's papers, and because he's also been looking at how old these sponges might be. I believe it's Joe's group who made some estimates about how old these barrel sponges are.
Okay, can I, you moving yet or not yet?
Probably in another minute or so.
So what we're doing right now is the ship is moving. So what happens is the ship
Following the same
moves
rate.
and then sits still for a little bit. Sirius moves. D2 is able to move, and then we get to a point where we have to do that all over again. We're looking at a candelabra type of coral.
These are really pretty. Yeah. Oh, look at all
And
the
there's another-
nodules
I know,
on
look
the
at that.
left side.
I know,
My goodness,
I
I,
know.
just said that there were fewer nodules, and here we are back
There we are.
again.
Yep. And it looks like, thinking that might, well, first I thought that was a holothurian, but now I'm thinking that it might be a sponge. I'm not sure.
Mm-hmm.
Yep. Tara's saying sponge. I think you're
On
right. I
the
think you're
seafloor
right,
below
Tara.
the
Yeah, I think
lasers.
you're. Yeah, I think you're right, Tara.
Quick zoom.
Yeah,
Yeah.
quick zoom in on
To
that.
the
just
right
confirm.
of the
Yeah,
lasers
right
and below
right
at
below.
4 o'clock.
Okay, so
Yeah.
Oh, it's a spongy.
Yep.
All right, I'm
Spongy.
moving now.
Wow.
Okay, good.
Is that sort of?
Oh,
a,
and it
sort
looks
of a
like there
cup,
might be a. It looks like there might be a little cup coral.
Look at those
Is
lasers
that-
on the
Yeah.
port manip.
a cup coral.
Uh-huh.
Oh, wow, yeah.
The white.
They're like turning.
Yeah.
Okay.
Yeah, because of the dust.
Which is funny because you're only
We're kicking up some of the sediment with
Well,
the
yeah.
Oh, did you come down hard
wash
then?
from the propellers on
I
the
backed
ROV.
up.
That's why you see a
Oh,
little
maybe.
bit more cloudy view in front of us.
Yeah. Okay.
Interesting texture on these mounds of manganese encrusted rocks on the seafloor. Looks very similar to almost to small individual nodules forming on the surfaces of the rocks. It gives it a sort of
it
nubbly
does.
surface.
You know what? It does, Patty.
Mm-hmm. It was so fascinating. A few days ago, we saw a very peculiar holothurian. As we were moving along the seafloor, we zoomed in on what
All right, ready
looked
to push
like
up?
Okay.
a pile of rocks, such as those we see on the very right-hand side of
yeah.
the screen. And it turned out it was a holothurian that had picked up and used a bunch of the pebbles in the surrounding terrain to mantle itself. Little pebbles
itself.
and very tiny foram tests. So it had little tiny white specks and larger rocks just coating its body. I was moving along the seafloor.
All right, so we're starting to see some more whip corals.
And more individual rocks covered with manganese.
Yeah, Tara Luke said it's almost like a cobblestone street. Indeed,
might
it
need to make
is.
that
Well, that's a
Time
good
between moves a
analogy.
little less, Nev. Maybe
A
just
little
wait.
less?
Yeah, just maybe wait like just like 2 minutes or so.
Between the end of the last one and this one?
Yeah.
Mhm.
Oh, okay. Yeah, then I'll put it in here in about 1 minute.
Sounds good.
Okay, looks like a black coral over to our left. Some more whip corals and candelabra-like corals.
What's your, it's good.
Yeah. Too late.
That's right. You'll get a feel for it when you see the bump.
Ah, Here we see sediment without nodules. Tiny patches.
Bridge, Nav. Same move, please.
There are few
having-
things here.
Yes, there are.
Growing on the rocks.
We are at 1,795 meters.
Great.
We're seeing some hexactinellid sponges, a couple different types. There's a stalked one that we just went over. Yeah, yep.
Guys,
Is it,
could we take a look at the base of that weird vase sponge that's up to the upper right?
Pilot, watch
that
the.
might really help with an
We want to
idea on
take a
that
look at
strange
the
one,
base
since
of
you
that
can clearly see the
sponge
base
to
right
the
now.
upper right.
Upper right. This white stalked one here is what you're talking about.
yeah, roger that.
Okay.
Vase
Okay,
sponge.
will do. Yeah, Chris, this will give us a better view of the base of it. This is one that we saw earlier and imaged earlier. We got a good image of the top of it, but I think the base was hidden. So, yeah, this will give us a good image of the base.
Yeah, the reason being is that there are some bases that are in the Corbitellinae, in the genus Corbitella, that actually have the fibrous connections. So they're kind of weird euplectellids in that they're lophophytous. And this could tell us whether it's one of those or whether it's a Regadrella or a Dictyalis.
Yeah.
And for the
Only
people
gonna move for
listening
another
and watching from home,
30
can you
seconds to a
explain
minute,
again
and then
to
we'll
us
have,
what
like, 3 minutes of,
lophophytus means?
Pause time, so we got time.
Sure. So these sponges attach to rocks or to the sediment in two different ways. One way they do this is to glue themselves onto the rocks by secreting a siliceous plate, and that's very, very
I'm
obvious
just
when you
stopping
see
that last
that.
move. Okay.
The other
And then
way
just
they
3 minutes
attach to
between-
rocks, and you see this in sediment-dwelling sponges too,
started moving
is
again
they
when you
have a
plug
very
in.
fibrous
It'll be
base
about a 3
to
minute
them, and they
delay
kind of
until they
anchor
start
themselves down, and that's
So,
called
48.
lophophytus.
So this one looks like it's not lophophytus. Would you agree? We're going to get a little bit, going to try and zoom in a little bit closer if we can. Not sure if we can.
Can you zoom in more? Is that okay?
Yep.
I agree with you, Shirley. I think it would be more obvious at this point yeah, it's looking like a plate to me.
To me also, Chris. Very nice. Wonder if we can
Yeah, so
gradually
that
work
would
our way
rule
up.
out the genus Corbitella. And so that was actually very helpful. So then this is probably either a Regadrella. My guess would be it's a type of Regadrella. I guess. It's looking pretty weird.
It's
though.
looking pretty weird to me too.
Strange
Yeah,
Regadrella,
yeah,
though.
how the tissue kind of like swirls around. It's
Yeah.
almost making kind of like a
And a-
spiral.
and a meshwork
but
at the
see, it's got
top.
that euplectellid type sieve plate at the top.
Yeah, so they're both are in the euplectellid, and then this one has a more substantial sieve plate, which is all of that sort of mesh that's right on the top of the osculum or hole in the top, than the Dictyalis that we saw the other day. Also, it's got these ridges running down the body of the sponge, which is very different. So I still think Regadrella is the best fit for this for now.
Wonderful. Thank you. Thank you, pilot.
Okay. Moving on.
Boy. And the boulder it's sitting on looks interesting to me. Or wait a minute. Nope, it's a different boulder. Gosh, I was looking down to make notes, and suddenly there's something new.
Something new. You
Yeah.
can't take your eyes off the screen.
No, it
for
changes.
a-
But this huge boulder
Second.
looks so much more angular than
Yeah.
anything else that we're seeing on the screen. It looks like it's turned over and exposed a fresher, unmanganese coated
Okay, so
surface.
let's see if we can look at that one on the left-hand side. The fan on the left-hand
Okay,
side.
roger that.
Yeah, I'm assuming you're
No.
the one you want to look at
You
is the
gotta
one
be
on the kind of
It's about to get a
left-hand
little
side
bit
of the
steeper.
screen right now.
Coming into
I'd like to
center,
not
Coming
hang
into
out for too
center.
long, just
Okay.
a little above
just be
center.
safe.
Is that right? There are some other
It's not like
branching
Not too much of a
Corals
concern,
to
but just better to be
a
stretched
little
out
lower
than-
down the slope.
No,
Be
that's not
surprised.
what I think he said. Back left, so I just wanted to double check.
Is this
Sure.
the one center screen?
Just trying to verify.
Okay, roger that.
I would say go ahead and do a zoom on it, though, please.
Okay,
Yeah, we're moving now.
okay. Video, you want to do a quick zoom on that?
I got a lot of visual and serious on that last heave, so
Holding push.
I think we'll be okay.
Ah, sorry.
Okay, so Les, you're saying more to the left. Are you talking about this one we're zooming in on now, or another one?
Yeah, that's what I was wondering.
Yeah,
there's
I
some
can't
there's
tell.
some down the slope a little
No,
bit.
there's lots down the
So
slope.
we're just, yeah, we
Well, I
just
saw
started that.
two
that we
Yeah.
called down.
specific
So we need
ones.
to get moving?
Yeah, let's push out.
Okay.
Yeah, let's push out because I think they want to
And
look. I'm not sure if
Just
they want to look at the
rotate slightly to
Antikythera.
port.
We need to push
Yeah,
out.
pilot.
We got to go
If you
pilot.
can, yeah, look at
Roger
those two. If it's possible to get
Yeah, I
back
think we have
down
to
there.
move.
No,
I think we
it's-
have
it's
to
too
move. We
late.
have
Watch
to
it.
move.
Yeah, we need to move, and there is
Oh, well.
some steep terrain
Yeah,
right
let's
ahead.
go.
That's
Let's
the
go.
challenge
Yeah.
with using the system. Is
Yep.
that we have the ship on the surface and a cable
Ooh, look at that.
down to
I don't
the.
think it's very high, but
Yeah. Shirley's saying, Patty, talk about the rocks.
Oh, look at
we
the
are
rocks.
looking
We're
at
coming on
a
a
very
Let's stop the ship.
vertical wall.
We are.
Bridge nav.
Oh, cool. I like the fact that it
We
has
like
little
walls.
rocks at the bottom, yes. Well,
Hey,
walls
we're
always
done with that last
expose
move, aren't we?
things that are
Okay.
in place.
Okay. Then we're good.
It doesn't
Which is
look like
cool.
it's that.
No,
yeah,
let's
there's
hold
not a whole lot.
so we can hang out
Looks
here
like
for a little bit.
we can hang
No,
out
it's not a
here.
very tall wall, but it is a wall.
Yeah, you can hang out for a little. So we started moving at 48.
So
we got
it's
about 3
about,
minutes of movement.
A little
But
bit,
we'll be okay. So if
little
you
bit
wanna hang
bigger
out here
than
for
the
like a
height
minute
of the
or two.
vehicle. The
Okay.
vehicle's about 9
Get a
feet,
zoom
so
or
this
two.
is 3 meters or so.
I just figured they'll want to look at this.
And the overhang at the top looks like it might be
Crust.
part of the
Yeah.
manganese crust.
And if you want to port lateral, just to stay in center view.
So that gives us an
Just a
idea,
no pilot,
too,
your
about
swing
how
arms
thick
are
that
out
manganese
lower.
is.
Roger
Although
that,
all
thank
of
you.
the slope that we're looking at, despite the fact that it is rougher, is still covered with manganese. It's not as thick, and that is why we can see this rougher surface texture. And
So-
of course, we have critters hanging on the
Yeah, a
wall.
lot of the fans that we've been seeing are this Candidella.
It's a cool ledge,
It's
isn't
a
it? Is that just a
primnoid.
manganese ledge?
Oh,
Are
man.
we seeing stalked crinoids in the background with the feathery tops?
We
Okay.
are. We're seeing some black coral, so those kind of feathery type.
And cup corals over
Cup
on
coral.
the right, small one.
Yeah.
And behind it is what's that guy? The fuzzy guy behind it.
Chrysogorgia.
Chrysogorgia, right? Oh, we have a holothurian crawling up under the ledge.
Under the ledge.
And a sponge down there. A lot of things. These critters do like ledges,
So we got a little bit
where
of
the
time.
currents are stronger
Just another
and
minute
there's
of
some
movement
protection.
or so.
Accumulation of those dead stalks. Sorry, stalks
this
of
is
dead
at-
sponges.
Uh-huh. Indeed.
Starfish up top there, top
There
left.
is Sea Star up at the top. So we're waiting for the ship to stop moving, and then we'll get a
Ship-
little bit closer look at
ship
some
should
of
be
these
stopped now.
organisms. We are at
Or coming to a stop.
1,785, yeah,
80s. Yeah.
86 feet,
Well,
meters.
yeah.
Is there something in particular you want to look at out of all these choices here?
The pilot is asking if there's something in particular that we would like to look at out of all of these choices that we've got right here.
Yeah, I want to see an open surface of a rock, pilot.
Okay.
If only.
Yeah.
Sorry, we aren't going to see those. Got crinoids
Hey,
hanging
Watchley,
on for dear
Do you
life at
think
the
hanging
edge of
out
this
here
overhang.
for 4 or 5 minutes is going to be enough time? Or do you want to stay?
maybe a little bit longer.
A little bit longer. Okay, we'll hold
Yeah.
off on that move then, Nev.
Yeah. Okay,
Copy that.
shall we come up over that ledge? You want to come
Sure.
up over the ledge?
Sure. It looks like there's a drool of something coming down this ledge. This looks, the
drool of something.
morphology looks very pillow basalt-like.
I'm
However,
going to turn my head to follow you a bit,
Okay.
but we are off track once I do that, so just keep that in mind. Not that we're moving.
Watch
but
lead. Were you talking to me? Okay. Okay, video, can you zoom in a little on that?
That's good. It's- we can move along that ledge, please.
Okay, roger that.
If it's possible to keep just a slightly more of a zoom in on there, so that when we see things that are smaller that are along that ledge, we can make a choice
Snap
to
zoom?
snap zoom. Yeah, so
Okay.
just zoom in just a little bit more, just so we can get a closer view of what's on that ledge.
okay to zoom out.
Yeah, go ahead, Tom.
That's good.
That was a little sponge to the right.
Just about out of there. Yeah, I'll have to start working back the other way. Yeah, I see it. Yeah. No. Yes.
With the view.
Yes, I believe so.
Oh, look at all the stalks down here, covered with manganese. There's a
Holothurian.
holothurian.
Oh
and
my
a
gosh,
sponge.
look at all that stuff. Okay, let's take a look at that.
Pretty.
Come up,
Coming
It's a
up.
garden.
up a little bit, please. Oh, wow. Oh my gosh. Oh, that's
That's a spiral.
Iridogorgia, I think. Wait.
Yep.
Okay. Oh boy. Iridogorgia, Scott agrees with you.
Yay.
Magnus spiralis says
Oh my
Les.
gosh, it is. Look at this.
It certainly
Okay.
is Magnus.
And spiralis.
Yes, definitely spiralis. Ooh, and the rocks are cool too. Lumpy rocks are much more fun.
Go ahead.
Wow.
Okay.
It's interesting.
Oh, it looks like that. Is that a metallogorgia over on the? It's out of the view
I know
now.
what you're saying. It's on the right,
Yeah, it's on the right-hand side.
and it has that
Like they-
flat,
What you were looking at.
flat surface.
Yeah, let's get it closer.
I'm
Let's
not
get-
sure it is.
Yeah, it could also be that black
Yes.
coral
Okay.
Belopathes as well.
Yeah,
The
we saw
one's
before.
The highest point there, right? That's what you're looking at. Coming in
You
on-
know, what we're looking at here is the edge of scarp that fell away down slope. We see lots and lots of organisms up here on the top, and what we see immediately below the scarp, or the overhang that we were looking at, is lots and lots of stalks covered by manganese, which suggests that when the stuff fell off, the critters were no longer happy. So the thing at which the lasers are pointing, almost, yeah, just to the left of the lasers, is what we're trying to take a look at. I think they've been turned off now. No, they were on a
See the
moment
lasers,
ago.
okay.
They were on the back
Okay.
of this.
Okay. All right, pilot, can we zoom in on that when you get settled?
Yep. Go ahead, Tar. Zoom in on that. Let's see what we got.
What is it? Oh, you know what? Does look like a Metallogorgia, I think.
Okay.
I think.
Go in.
Yeah, so Shirley,
I'll
this
hold
is
for a second.
Metallogorgia. You nailed it.
Yeah, yeah, yeah.
You go for a bit closer zoom.
I got it, I think.
So this is also a Chrysogorgiid. Is that correct?
Yeah, that's correct.
I think it's
Les
fascinating.
Watling says it's a
Still,
very
we actually
old and
are
mature
the exact same.
Metallogorgia.
Okay.
So
That's
we still have
good.
to average 0.1.
So we've been holding our average.
Yep.
Asako is getting a lot of pixelation, she says. So she may not be able to see this beautiful, beautiful view.
Just gonna hold
You
here.
want to get
again.
rid of the lasers now, too?
Sure.
Yeah.
Lasers off.
Anybody else seeing pixelation?
Okay, watch leads. Is that enough? Or
That's good.
Very
Thank you,
good.
pilot.
okay, anything else
All
up
right.
there? Since we're
Not
sitting
sure.
here.
Do we have time for a quick
Okay,
establishing?
shore-based scientists.
yes.
Anything else that you'd like to take a look at up here? A closer look while we're here before the ship has to move again.
All right.
Okay,
Clear.
clear. All right, watch
Awesome.
leads, so we're moving.
Okay, we are moving.
All right, so we got a long way to work back to starboard.
Yep.
All right. Let's do it. Let's go ahead and call in the 30-meter move. We'll be lined up by the
Bridge
time we
nav.
start moving.
Hey, guess what I want? I do. You're good.
So again, seeing some fan-shaped octocorals. We're seeing stalked glass sponges. This flat surface here, relatively flat surface, still manganese crusted. Oh my gosh, look at that big rock. And coming on this rock pile. Patty is busy scribbling
Sirius is
down
lined
notes in her notebook.
Okay.
We have another Walteria in the background. We have
We got
branching
3-4 minutes before we move,
coral,
so
and we
got
have
plenty
that
of time.
beautiful bushy, probably a Chrysogorgiid.
We're seeing a number of fractures on the seafloor. They're all sort of oriented upper left to lower right, which would be consistent with approaching the edge of the slope that is
what are those
oriented
cracks?
upper
Oh.
left to lower right along the track. And now we are seeing them
these are
because we are
cracks.
oriented right along
Ooh.
the edge of
cracks.
the fault, and
Lots
these
of fractures.
these fractures are all associated with the fact that the hillside fell away to the southeast, right where we are. So these are the kinds of things when you are approaching a very steep fault scarp that you want to stay behind. In other words, we would like to be standing to the left here rather than to the right with the cracks behind us.
So is this something that when this happened,
whenever.
that would have caused kind of a massive die-off of some of these
Well,
sponges.
that's what I was
for which
thinking
we're
when we
seeing-
were looking at the overhang, because
Yeah.
we saw all the biology alive on top and all the stalks below. And what I'm thinking is, as things fell off,
All right, edge of your tether.
the rocks were jiggled
Okay.
about,
That's
the
very
animals
cool.
fell off,
We got enough to make this boulder
got
here?
broken
Maybe.
off
We're going to be moving in
the
a minute or
seafloor,
two.
Okay. So
and
I'll just
accumulated
be there
as
soon.
dead
Slowly
stalks down
sneak
below,
up on
which
it.
is what I was scribbling down in my notes
Oh,
here, so I
good.
can write it up for later.
Good.
Yeah, you are pulling.
And
right
these
Okay.
boulders must have come from someplace else, because the whole seafloor is very flat and lumpy, covered
come
by
back
this
and lateral to port.
manganese coating, and yet we have detached large boulders sitting on the surface.
now. That's the trick.
Copy that. Who's this guy here?
Checking out that circular one.
Yeah.
I don't know what that is.
What are you looking at? That curly Q?
Yeah, curly
A
Q
whip. It
center
looks like it's a
string,
whip.
Just a whip.
Yeah.
It's pretty deep crevice. Little crack there.
Yeah, it really is. Can't see down into it, or not too far.
Oh, wow. Look at that fracture.
Ooh, definitely we want to be on the
A
left
crevasse.
side. Well, a mini crevasse. I wonder if we could. Co-pilot watch lead. May we have the lasers on for a
Pilot,
moment?
lasers? Would you like lasers on, pilot?
Yes, please.
We'll get lasers
Sorry.
on so that we can take a look at how wide that crack is, maybe. Okay, two little tiny dots, 10 centimeters apart. And so we're looking at a crack that is 20 to 30 or 40 centimeters wide. So maximum of maybe 16 inches across there, more like a foot. Easy to hop over, but if it's moving down slope fast, it might
You
get
better
more
run fast.
and more
You
difficult.
better run fast.
Right.
And if you're attached, forget it.
Okay.
Yeah, for sure.
And many of these organisms that we see here
Look
are,
at the
in
view in
fact-
Sirius of those cracks.
Oh, yeah.
Ooh, that's impressive.
Okay, we have a long whip coral here.
Okay, we should
Get a
start
snap
moving if
zoom
we're
of
not
that.
Yeah, we're moving a little bit.
Pilot, can we get a snap zoom of that?
Sure. Video, go ahead.
Looks bamboo.
It does.
It is.
Ooh, Scott, are you oohing and awing?
Okay.
Scott, I
Is
think,
that
is
good?
having dinner, but Les is on the
Oh,
line.
Okay. I'm finally warming up
How's
here.
that watch lead? Enough?
That's good, thank you.
Okay, moving on.
All right, let's push out.
Pushing out.
Looks like we might have another little
Oh,
bit
there's
of
another
a
oasis up
cliff
there.
face
On
and
the
plateau
boulder.
up ahead of us.
Yeah.
Okay.
They do like the boulders.
Yep.
Maybe more interesting stuff.
Yep. Pilot, if we can move towards that boulder.
Going towards the boulder. Roger, copy.
Thank you.
You're welcome.
And it'll be just about
big
time for
cracks.
another move in about a minute or so here.
Okay.
Shall we keep up that pace, or do you want to wait based on what we see up
let's
ahead?
wait on, yeah, because we got another maybe interesting cliff face on the sonar.
Okay.
It's, there's a white thing up to the upper left that may be interesting later after this boulder. It's like a big sponge.
I
Yeah,
keep
you're not gonna have a
wondering
whole lot of
where
time
these
here.
boulders are
Okay,
coming from
Roger,
with the
copy.
thick incrustation of sessile biological things. There's no,
Lots of stuff,
there's
okay.
no big slope above us.
Okay, so watch lead pilot.
Go ahead.
Yeah, we'll want to take a look at some of the things on this rock.
All right.
Like the
we
golden-
Yeah, the
are
golden
moving,
one.
though, so we don't have a whole lot.
Okay,
of
let's
time.
do a snap zoom on the golden one,
Okay.
please.
One dead center there, roughly. Okay, video, go ahead.
Okay, holding partial.
All right. It's finally
Shirley, what is the more delicate looking thing right in the front?
Oh gosh,
to
yeah,
the
that
right?
purplish one. Yeah. Okay, so
We've seen that before, but I do not remember what it is. But it's very pretty.
Okay, so plexaurid.
All right.
Mm.
Got another
Thank
20
you.
seconds or so.
Scott,
I'm going to take the closer zoom so I can tell what it is.
And I'm going to need
Our
you
ship
to...
And the
isn't moving now.
Chrysogorgia.
So I think we'll
And
have about
Asako
3 more
says,
minutes of 2 more minutes of
okay.
movement.
We started
I'm
at...
reading all the biological
We start at
comments.
106.
Anybody want to
okay.
describe
Yeah, you're right,
this
yeah.
for us as we scan it?
It's only 2 or
with
3 more
the
minutes to
video?
swing, so... But I don't want to get in the habit of...
That's good,
Getting
pilot.
the
Thank
yeah,
you.
Okay,
right.
roger that.
Sorry, go ahead.
Especially,
Okay.
you know, sonar
Roger,
returns
we're
like
gonna-
that. Not that it's going to be very tall, but it's just good
I'm
practice
eating
to
his dinner, so I'll do it.
Get a look at that,
So
white
the
sponge over
plexaurid
to the
is
left
sort of
if we have time.
then I think we got to push out.
a very flexible sea fan, if you will. And
Around this side
that's
of
one
the
of the
boat.
ways that it differs from, say, the hard scleractinian corals that we've seen, and also in the corallid, the hard precious coral group as well. One of the characteristics, now that we moved off of it, you can't really see it, but one of the characteristics is they have a lot of scales on their branches, and that's one way we can sort of diagnose them when we see a close-up like that. But also now I think most people can spot them from a distance. They just develop a gestalt for what a plexaurid is versus some of the other types of gorgonians down here.
I'm still working on that. Okay, so now we're in an area where, again, not much biology. I shouldn't say that. It's relative to what we've been seeing before.
Good there.
Yep.
Roger's good.
Yeah, he'll get a feel for it.
Okay.
Just got to let it happen.
I've tipped
Deb and
quite a
I've
few
been
rocks in my day.
wondering about these boulders. And I now, having seen all these fractures that are parallel to the slope, I'm wondering if there are steps to our left, further toward the middle of the slope, that are shedding these things down onto blocks that are stair-stepping down
Okay, we can get another
toward
move.
the really steep edge of the cliff that we're running
Bridge,
along.
Nav.
So there may be some terrain that's less deep just to our left.
Let's get another move. Same one.
But how the critters could remain on top
Okay, you
of
got
them,
some
then
time
have
now.
to
Okay.
slid right down, you know.
So I would kind of move
That's the
laterally
noise
here.
rock sliding makes.
It wasn't nearly as interesting of a feature as it looked like at first.
Without disturbing
looks like there's
the biology on them. But we are seeing a lot of stalks on the
Yeah.
seafloor around here, so
Right,
something
and-
is disrupted.
Right, and then over on the
That's a long
the
shot.
left-hand
you know.
side in
Yeah.
the back,
You gotta
there's
back
a
up a bit.
very long whip. Very long
Ooh,
whip
yes,
in the
there
back.
is. In front of the field of view.
Yeah, we're at the end of our leash right at the moment.
Okay, we're at
so-
the end of our of the tether right now, so
Uh-huh.
we
Gotta move.
actually have to sit still.
While the ship moves.
No, don't come back too fast here.
Okay.
Because this shows relative angle between Sirius and the ROV. It's way ahead of the
Yep.
ROV. Doesn't look like it on the Sirius
No,
view,
it doesn't.
though.
No.
I'm confused. Ooh, big
One of those
fisher.
sponges that might be. Oh yeah. I didn't see the crack because I was looking at the sponge.
Of course you were. That's your job. My job is to look at the cracks. Oh, more of those little.
So right now your camera's pointed up at
vase-type
14 degrees or
sponges.
so.
Well, so much more my theory that there might be a block of stuff shallower off to the left of the ROV, because we're moving toward that direction, and I don't see anything that is higher than this terrain.
So you called them to move at 110? Is that right?
So, ooh, crack
111.
all the way through the center of the screen.
Okay.
And there's a little shelf, little step break with the left-hand
another
side
crack
is
in it.
falling
Off the
down.
side of my
Yeah.
screen?
Wow.
Okay.
Okay, well, that's downslope, so that's not too surprising. It really is sort of fascinating when you look at the more detailed bathymetry that we'll pick up in an area like this overnight. And you see so much more structure. And then when you get down on the seafloor with the ROV, it's like it's exponentially more complex. So it's kind of a fractal pattern. Shirley, that
Look at that.
is that one of these basket sponges leftover bits? Or is that just a rock?
No, I think it's
okay.
I think it's
just
a rock.
I'm sorry.
It's a
caught
rock.
me. You
It
caught
is
me
a rock.
doing the just a rock thing. You can fire me now.
So
I can just go hang out in the lounge.
and work on the cruise report.
No, watch movies. Are you kidding? If I get fired, I have to watch movies. I haven't seen a movie this entire cruise. Oh, that's not true. I say one half of one movie.
Okay, so again, seeing more of these
Ooh,
fan
boulders.
corals
And boulders.
and boulders. Yep. We have about 2 hours and 15 minutes left in this dive. We are at 1,771 meters, and our target depth, stand by, is 1,679. So we're doing pretty well. We have about 100 meters vertically, you know, that we need to traverse,
I'm
but
moving.
Uh-huh.
now tells us we'll do it.
Okay, we're seeing lots of big slabs that have broken apart from one another. In some of these there's almost a jigsaw puzzle type fit. So these are broken off bits of the maybe even just the manganese incrustation.
Right at the edge of that cliff.
Would have
That's
a
cool.
different. Oh yeah, I see the sea star.
Yeah.
Different texture
Yes.
than the top of the rocks beneath them,
Move
and therefore
after
can
this
more
now.
readily break
The
off
next one
and
we put in, we'll
slide
probably have
toward
to
the
adjust here.
edge of
Copy
this
that.
incredibly steep slope.
There should be some interesting stuff right at this edge.
Well, I wonder if this is a corallium, this fan over here on the right. I wonder if
This
we
is
could take a
cool,
quick
huh?
look. It's pretty dense pink colored, so I think it is.
Copy.
Oh, and look up ahead. There's a huge.
Oh my
mass
God, there's a
of-
gigantic
Is that
fan.
a-
Holy
Is
Is
no,
that a fan or a basket star?
Not sure, Don.
No, it's not
sorry.
a basket.
No. So
The pink one.
we're gonna look. Yeah, we're gonna look at the pink one
Pink
first.
one.
Because
So
it might be
actually,
a
we're going to feel that
corallium.
move
So
starting
is the
about now.
white-
Then we're going to, yes, part of
Oh,
the,
man.
So we'll
and
kind of be
then,
skirting
I don't
along
know
the
if you
edge of
guys
this.
can see it, but
Okay.
in the background
But I'm
there is
pretty much, yeah,
a
lined up with our direction of motion,
ginormous
so-
fan.
Just keep
And
an eye
don't
on
land
yourself
on any
and
of these
serious.
rocks because
Right.
you're on the right side of the crack,
This is cool,
and
though,
it could
isn't
go
it?
down the slope.
What was that noise?
Oh, that's a big breaking. That's
So let's try to stay
okay. The
above
white
it and on top
bottom
of it as much as
this
possible.
is that bleached, like
I'm
the
moving
kind of
now.
thing we're seeing on corals all
No.
over the world.
No,
It's
because
just
that's
something
completely
else?
different. These
Okay.
corals
Not much
don't
time.
have
Okay, Roger.
the dinoflagellate symbionts, the microalgal symbionts that are expelled from the corals when
When
it
they're
gets too
bleached.
hot. Yes. Now
Aha.
these do not have those symbiotic zooxanthellae in them. So no, my guess.
These corals are, however, doomed.
Yeah.
Okay,
They're
video,
doomed.
They're
just
going to
gonna
go-
turn a little bit. Zoom in.
Roger. Okay, zoom partial.
Yes. Yes, please.
Let's
Yeah,
stop
we
it
would
now.
like a
Stop
zoom,
moving.
if
Okay.
Zoom
possible.
partial.
Yeah, go ahead. Partial. Yeah.
Roger.
Move back.
Okay, I'll hold here for a
Ooh,
minute, and then I'll go
nice
in
brittle
further.
beautiful
Okay.
Cora.
top.
Yeah, this is a
Oh,
corallidae,
Stop
There's
this
also
here.
what are they called?
crinoid.
Thank you. A crinoid on the backside. A
Beautiful.
yellow
I
crinoid.
think
So-
I'll stop.
zooming
Yeah, go ahead, Chris.
This is a good one. We certainly haven't seen very many of these deep. It's actually probably the genus Hemicorallium in the family Corallidae. We just haven't seen a whole lot of these in the Marianas in general, so it's really good to come across them at least occasionally, so we know they're there.
Is this one of the precious corals?
Yeah, it
we
is
gotta
precious
go
coral.
right
It's
in.
awfully deep, so I doubt if anyone would attempt to try to harvest it for jewelry making or anything. But it's got that hard skeleton, and it's got the pink color, and it's a member in the precious coral family,
And I'm going to
Yeah,
come
we
in on
gotta
the
do it
top.
right
So
now.
a bit
So
further
what
What
down.
is growing on the bottom of it, Chris, in the white?
That's just the skeleton.
If you look at it, there are
bit
larger
down.
tentacles coming
Yes.
out of the polyps.
Thank you.
Could you tilt down?
We need,
Tilt
to,
down.
we need to
Yeah,
scan down.
I might have missed what you were talking about. I know it's whiter on the
Okay, hold
bottom,
on a
and there's
second, watch
plenty of cases
No,
where
look, it's
the
different.
corallids have
Tara,
different
when you're ready,
colors.
then we'll look
Like
at
you can
the base.
get color in
Okay,
toward the end of the
stand
branches,
Okay.
or even color in the main
Yes.
branches, and then white
Copy
tips.
copy that.
So not really sure if these are just whiter polyps down here. What's going on there?
Okay.
Okay.
Yep.
All right.
You
going
wanna
down.
tilt
Chris,
down
at
to
the
the
base
Yep.
of this, you'll see
Going down
much,
to the
much
base.
larger polyps.
Oh.
Is this the same, or is this something that's encrusting this? We're at the bottom now. Should see it shortly.
Yeah, I'm seeing it, and I'm thinking about it. I wonder if this is some type of paragorgia or something
Yeah.
else. I don't think it's the same thing, but I don't
Yeah.
know. Could be. I don't know. Maybe Les or Scott can weigh in on this.
Yeah,
zoom.
Scott, tell us what's going on, Les.
Oh, and you see it's encrusting over the surface of the
yeah,
rock, too.
yeah. It's
Zoanthus,
encrusting.
maybe? Or, I don't know.
Something else.
No, it's not going to be zoanthus. Oh,
Well,
I
it's
don't
not
know.
a zoanthus.
It's not a
It's an
zoanthus.
octocoral.
Yeah, it is
so-
an octocoral.
So
Yep.
it would be a
Stoloniferous,
stoloniferous,
yeah.
in that case, if it was growing over it.
Could we get an establishing?
Okay, so
So Asako Matsumoto is putting in a request about collecting this. Could be a pretty interesting collection if everyone agreed.
So what do you think?
I would
turn
like
off
to
the
hear
lasers?
from
The
Scott
rest?
and
Yeah.
Les.
Thank you.
Please, because we've got whatever
And
might be in the
Asako.
background, and Asako,
Yeah,
yes, as
there
well.
are quite a lot of other animals
All right, since I'm
sticking
here,
out
I'm
from
gonna
this.
zoom.
We're
Just
right
let
at
us
the edge
know.
of
And
the
they're
steep
waiting for a
scarp.
minute.
Let us know, but let us know quickly.
Zoom on the transition where it turns from
Yeah, and the
white to
tricky thing about collecting these corallids is they're so brittle.
Brittle, very brittle.
Those that
Yeah.
grab it, they'll probably just break it up. It could just fall right over the cliff face
Tell
there.
Bridge we found something really cool.
Bridge, we found something really cool, so we had to back up there.
How much more time do we have, Shirley?
It's very neat.
We have about 2
Okay,
hours
video's
left.
clear.
Whoa, fantastic.
What are we thinking, folks? Yes, no?
Well, people are saying it'd be worth a try to collect it. It's a pretty unusual colony here. What's going on there?
Okay.
I could give it a try. I mean, I would vote for trying for it.
So what we would like to have on this is one of the branches that has both pink and white, please.
Okay, I think
Copy that?
And it's very
So-
brittle.
just that lower. We could try to snip.
Yeah, I
That
think
lower
that's what
right
everybody
branch.
would like to see.
Yeah, it's very brittle.
Very brittle. Okay.
Yeah, likely to break
right.
up.
okay.
That's what I was doing.
And Bridge
Sorry.
Nav, we can go ahead and hold here if we're still moving. We can hold here if we're still moving.
Just warning, this could get ugly.
Yeah, understood,
thanks.
Chris. We're having a little musical
Okay,
chairs
let's
thing
see.
here in the front row.
Okay.
We're thinner.
All right, so we are
All
going
right.
to go for this. We understand that this is definitely going to be brittle. We're interested in the part on the
right,
right-hand
we
side
have
that has
something
both
to report
red
here right
and
now.
white on it. Dan?
And my understanding also was that they didn't want to open that box because it's a floater, correct?
Yeah, we can try
Yeah,
for the starboard bio box.
Yeah, we want this to go in the starboard, one of the 2 starboard bio boxes. Yeah, so Dan, not sure if you copied. We want white and red,
ramped
please.
You know,
White and pink.
moving
Let's
the
bring in the
ROV
lower swing
around to
arms.
face
Okay,
the
roger
slope
that.
and get the box underneath the scarp.
Lower swing arm, okay. Left.
I know.
Don't forget that
Copy
the
that
thing in the
one.
other bio box may float.
Yeah, roger that. We just talked about that. Thank you very much. Really appreciate that reminder. We're gonna go for the starboard side.
You are. All cameras are.
Okay, the lower swing arms are in.
Okay.
And retracting the pins on the bio boxes. Good, and we're going to go for the starboard side. When
Yes,
you
the
get
starboard
starboard.
bio box.
So extending the pin on the starboard side.
Ooh, what is that? Is
Yeah,
that
they're
an
more-
anemone
they're
down
more on
below?
the-
We're ready to go.
Are those anemones?
No, they're more coralliums, or what did Chris call them? Hemicorallium.
Below?
Well, on the right-hand side, the lower right,
no,
there are more
I'm just
hemicorallium.
immediately below
Oh, I
where
don't
we're
know.
going to sample.
This is so cool.
No,
Look at
it
that.
isn't.
No,
Yeah.
nope.
So
Yeah,
much
she, you're
stuff
right,
up here.
Shirley. There's a bunch of them down there, but
Yeah.
they're all monocolor, right? So they're not getting overgrown, I gather.
Right, yeah.
Yep, it would appear that way.
Yeah, so if there's any way he could grab that second smaller
Definitely can.
branch there, halfway down it that's part white, the closest one down below the other one, then that might be easy enough to grab, and you might be able to get it pretty intact.
That's what we're
We've
headed for.
We've nudged him about it several times. I'm afraid to even ask him again.
Copy that watch lead. You can remind me as many times as you like.
You're so kind
Oh,
and
you're so sweet.
understanding.
Thank you.
Folks, if you look at the quad screen view, you'll see 3 images of what the pilot is going to be trying to collect, and you'll also see all of the rest
Oh,
of
my
the
gosh, look
biota
at-
on this incredibly steep scarp. We're
Yeah.
right at the edge
Soon to die, right?
of the block.
Right. In another 1,000 years.
I don't know. See,
I know.
this
I
area
know.
would catch that block when it falls down, and everything will tumble down the slope eventually.
It's like houses built on cliffs.
Yes, houses built on cliffs are doomed.
But for a while, they have a great view.
They have a great view, and even on the way down, they have a great view. Won't last, but if you don't want to sell, it's perfectly fine.
Okay. Hydraulics up.
Yep.
Wow, there's a lot of stuff stuck to these cliffs.
All right.
Chris, you're so right about the fact that these are good hunting
please.
grounds. And I'm wondering if we might find a rock.
Yeah, this is shaping up to be a pretty nice place, all right.
Yeah, you bet.
A little bit closer. So you wanted the red and the white, right, watch lead?
Of course.
Okay.
Or purple is good.
It's very brittle, is what I hear. Oh, oh.
It's going to be very brittle.
It is incredibly brittle.
All right, let's try this.
And those reattach, is that right?
I
Shirley?
have no idea.
I
Gotta get my
thought
fingers in
you
there
folks
somewhere.
said that
I don't
Yeah.
know.
they do tend to do that.
I don't know.
So, like, good luck.
Is that enough there?
I think so, as long as we have some of
Yeah.
the white.
Some of the white and some of the pink. Wow, it
These
really
are like
is
as brittle as a stylast.
Oh boy.
That's
You
all right.
got
That's
it.
all right. It's fine. It's
Oh,
okay.
excellent.
It's
It's a
okay.
lucky
That's
day.
good. That's
Very
good.
nice.
That's really good.
That is great.
Okay, take your
Way
camera.
to go.
Oh, right
Let's just
in the
get it in
bio
the
box
box.
immediately.
Yeah,
Okay,
yeah,
right.
yeah. We don't
what
need
we
any.
want-
We don't need any
I want you
twirling.
to
No
go
twirling.
forward, but keep a close eye on the port toe.
That was one of the
Because
best
you don't want the
collections
box
of a
pushes away.
corallium I've seen. Normally, again, they just go to pieces.
Fantastic.
Okay.
We'll relay that in a
So-
moment.
Clear. You want me to take your camera and tilt up?
Let's get Mini Zeus on the right monitor,
Okay.
and let's double check that the port bio box pin is retracted, and the starboard
The
bio
port. That's correct. I already did
So
that,
don't-
but I'll double check. There
don't
we
want
go.
their, other one to
And
float
the
away.
starboard one is extended.
Yep.
And okay, does that look good? We'll go ahead for open.
Yep, nice and slow.
Okay,
just in
drawer
case.
open. Here we go.
Nice and slow.
Moving.
And how does
So
that
the
look?
co-pilot is
Looks good.
slowly opening
Hold there.
the bio box
I'll stop.
sled, and the pilot
Yep.
will insert the sample
Just barely
into
touched.
one of the chambers. In the white box on the upper right-hand side of your screen, in your third feed, the feed to the right on the bottom of your video screen.
It's a pretty big sample, isn't it?
Yeah. The outboard box is the bigger one, correct? Or is it?